think of your favorite fruit. Let's take a grape, for example. Now imagine a bigger grape, or an even bigger grape. Now what if we can make our crops bigger, but also more nutritious, drought-tolerant, and even disease-resistant? Well, CRISPR, or clustered regularly interspaced short palindromic repeats, is a gene editing tool that can make these dreams a reality. Gene editing is known as the manipulation of genes through cutting, replacing, or inserting genetic sequences. CRISPR is a tool based on the immune defense system of bacteria and works by using the Cas enzyme found in bacteria along with a strand of guide RNA. Now, the guide RNA is used to locate a particular sequence on an organism's DNA and unwind it so the Cas enzyme can cut it. This cutting, known as gene knockout, inactivates the gene. Scientists can also perform the opposite effect, known as gene knock-in. Using the CRISPR editing tool, pre-designed DNA can be inserted by Cas into the cut made during gene knockout. With gene knockout and gene knock-in, agricultural scientists can make plants more nutritious, drought-tolerant, and disease-resistant. This would supply larger, more abundant, and more nutritious food sources while conserving farmland, water, and the need for pesticides that pollute our environment. So if you see a giant grape in your future, don't be scared, because you know CRISPR is at work.